question my lab is asking is, how can we predict the next steps a cancer will take and outsmart it?
How do changes in the structure and chemistry of DNA affect its function?
We want to know, can we understand proteins enough to generate the next generation of drugs that is protected against resistance?
The Cancer Research UK Cambridge Institute is one of the largest cancer research facilities in Europe, with investigators from more than 40 countries. We sit on the Cambridge Biomedical Campus, which is the largest biomedical research cluster in Europe. We're a department of the University of Cambridge, but a semi-autonomous research institute. Our work focuses on tumor ecology and evolution and spans the spectrum from basic science to clinical translation. Every year, we receive generous core support from Cancer Research UK. This enables us not only to provide direct funding to our investigators, but it's also enabled us to build and maintain a world-class research support infrastructure that really propels our science forward.
My question is, how do we turn a deep understanding of the mutational processes in high-grade serous ovarian cancer that cause huge genomic chaos into biomarkers that we can use for treatments for women in the clinic?
The question in my lab is, why are benign tumors benign?
The question my lab is asking is, how are immune responses regulated in cancer and infection? My lab is particularly interested in the hedgehog signaling pathway, and we found that this pathway controls key differentiation nodes during the anti-cancer response. As a junior group leader, I especially value the supportive and collaborative environment that we have here at CRUK Cambridge Institute. Being firmly embedded in the Cambridge science community, and in fact next door to Edinburgh's hospital, lays the foundation for outstanding basic and translational science.
The question my lab is trying to understand is how non-malignant cells promote pancreatic cancer and how we can stop them from doing so.
How do the relationships between different cells inside breast tumors affect treatment response?
I want to know why children develop cancer and how we can treat them with safer and more effective therapies. The Cancer Research UK funded Centre and Institute here in Cambridge work seamlessly together with the university to ensure that the breadth of clinical and basic research is brought to bear on the problem of cancer.
How do tissue-resident innate lymphoid cells regulate inflammation in cancer and normal tissues?
Why, when it contains so many mutations, aren't cancers of the colon more common?
How can we provide cancer patients and their treating physicians with more information to improve their treatment through the use of liquid biopsies? Blood samples from cancer patients contain small fragments of DNA originating from the tumor. By analyzing these fragments, we can find information about the tumor's genomic makeup and what's happening to the tumor in response to treatment. A few years ago, we spun out a company called Inivata, which has taken one of the technologies we developed and has now made that available as a commercial clinical test for patients.
One question my lab is addressing is, how can understanding resistance mechanisms to certain cancer drugs lead to more effective cancer treatments?
Here at the Cancer Research UK Cambridge Institute, we're searching for the best researchers from around the world. So what's your question?